So here I'm going to discuss some antiviral agents and some of the strategies for making antiviral agents, which is very different than making antibiotics to target your bacteria. So in general, this is just a picture of a cell and the different parts of the virus replicating inside of a cell. Now, typically, a virus is always going to, again, have to recognize, so this is the tropism of the virus. And so the recognition of some sort of receptor, which is going to lead to attachment of the virus. Then the virus is actually going to have to get from the outside into the cell, and that's referred to as penetration of the virus. And then after the virus gets through that first layer, it either is an envelope virus, which it can fuse with the plasma membrane, or it's a capsid virus, which it has to do some sort of uncoating and getting out into the cytoplasm. Now, after the uncoating, you're going to have the virus nucleic acid, and then it's going to do a few things. It's going to transcribe. So it's going to make more of the messenger RNA. It's going to replicate. It's going to make more of its genome. Again, it depends what type of virus nucleic acid it is. It also is going to do protein synthesis. Now, after it does all of these things in your cytoplasm, for the most part, it's going to have to try to make more virus particles. If it is a capsid virus, it's simply going to have an assembly, and it's going to lyse and release the cells. If it's an envelope virus, it's going to incorporate its proteins into the lipid bilayer of the host, and it's going to envelop its nucleic acid, and then you're going to get buddying of the virus particles. And so each of these stages can potentially be a target for your antivirals. So there also are, with envelope viruses, you can have attachment followed by fusion and entry of the genome into the cytoplasm directly. You don't have to go through this endosome. If you are a capsid, you have to go through the endosome, obviously. But if you're an envelope virus, you can go through the process of fusing with the plasma membrane. Now, one thing to recognize is that this is infecting our cell. So any sort of antiviral has to, one, be able to get into our cells, and two, have a higher affinity for killing the virus than killing our own cells to be effective. And so that's a very huge hurdle for antiviral medications. And oftentimes, the viruses have developed resistance to our immune system, and those same resistance are also helpful for resisting drugs that we develop to target the viruses. So here they list a few of the antivirals that are available and at what point they are trying to target the actual virus. So you have a few that are going to target uncoating of the virus. You have interferons or antisense oligomers that are going to target your transcription and your protein synthesis. Interferon will definitely affect that as well. Replication, this is probably the most abundant of the antiviral strategies, is to make what are called nucleotide analogs, and we'll talk about that in just a moment. And then another is our protease inhibitors. So these are going to be like for hepatitis C, influenza, and now SARS-CoV-2 are going to target that. Some can target fusion, but it's mostly going to be our nucleoside analogs and our protease inhibitors. Now, one thing to keep in mind that's not all viruses are treatable by drugs. We don't have drugs to target every virus. And we'll put SARS-CoV-2. Merck just made a very important breakthrough. Hopefully they will have a drug similar to a Tamiflu for SARS-CoV-2. That's what their data was suggesting anyways. So you can see here that we have a lot of the famous viruses, so you can kind of tell where the money is going. Obviously, it's the viruses that are most common, so common colds, even though they're called common colds. We clear them very quickly, so they're typically not going to be a target for antiviral treatments. It's more you're treating the symptoms after the cold virus is gone. And so with respect to treatable by drugs, we have our herpes viruses. We have our herpes simplex, varicella zoster, CMV, HIV, of course. AZT was that first drug for HIV, but now we have combination drug therapies for HIV. Influenza, Tamiflu is very famous. That has a very small window of time in which to administer it and be effective. We have respiratory syncytial virus, our hepatitis B and C, where we're going to have our protease inhibitors, and then we have some that target the papilloma and picornaviruses as well. So a nucleoside analog is, remember what a nucleotide is. We have our 5-carbon sugar. I'm going to try to draw this semi-good. All right, so here we have our ribose. Remember, this is our 2 position, and so if it has an OH, this is a ribose, and if it has just an H, this is our deoxyribose. And then this is the carbon where we have our base. I'll do that in a different color. So here's our base, and our base can be adenine, cytosine, guanine, etc. That's our bases attached there. And then we have our phosphates. Here we have our phosphates are going to be attached here, and then we'll have another sugar come in. And then here on our 3, we'll also have our other phosphate sugar. So another ribose or deoxyribose. So basically that is our sugar, our nucleoside portion of our sugar with the base. Obviously you're going to have the nucleoside complete. And so nucleoside analogs, their whole purpose is to replace the natural nucleosides going to be added to RNA or DNA, and by replacing that, they hope to become suicide inhibitors. They incorporate themselves into the genome of your viruses, and they don't allow elongation. No additions can be added to them. So they, in essence, truncate the genome of the viruses. So there are several nucleoside analogs, as you can see here. And so we have our green ones, which are going to be for herpes simplex viruses. And so we just circle these really quick. So here's a herpes simplex virus analog. We also have quite a few of them are for herpes. You can imagine it's a big target. This one right here, acyclovir, is also going to be good for VSV. So that's what that little star means. So VSV, you can use it for that as well. And then we also have terfidine, this guy right here. Now with respect to our CMV, so CMV is going to have a couple this guy here, this guy here. And then we're going to have some, of course, for HIV. So those are going to be our orange guys. We have quite a few of those. This is HIV, is orange. And then we have our more rare, I guess, Lassa fever, RSV. I need to make it internal, so it's for both of them there. It's not very purple, sorry. Let me pick purple. I didn't realize I put purple there. Here's purple, and then up here we have purple. Rhabdovirin. It's becoming more famous. So anyways, these are all going to be nucleoside analogs that can help target these particular pathogens. Now we also have different mechanisms of action. You have where you can terminate the DNA elongation. And examples of nucleoside analogs that will terminate, there's actually quite a few of those. So let me just circle these really fast. Oh, sorry, don't have the name. Almost this whole side over here. And a couple over here. And so again, by terminating, that means that they cannot elongate anymore. And so they basically are unable to make the genome of the virus. Now there are some that are going to cause mutations through sort of wiggling their way into the genome of the virus. There's a few of them that do that. This little bit here. And then there are others that are going to be specific for HIV through reverse transcriptase. Now, again, the idea behind all these is that your virus has to have incorporation of our nucleic acids into their growing genomes. And if you add an alternative to that, that blunts the addition of the nucleic acid to the genome, then that will prevent the virus from replicating. Now you might say that's going to affect us too, because how does the nucleoside know that there's a virus there? Why doesn't it have an effect on the rest of the body? And the answer to that is not as straightforward as possible. For some of them, it is very straightforward. So for some of them, you have a very clear reaction, and acyclovir an example of that. So how acyclovir does this is that it requires to be activated. So the nucleoside analog of acyclovir has to be activated in the presence of a viral thymidine kinase. And so this is why acyclovir is not as broadly protective, is because it requires an activation by a viral enzyme. And so if the virus is present, you're going to cause termination because acyclovir gets rid of that 3' OH. And if you remember on our picture here, this 3' OH is what is important for adding the next base pair to it, that next sugar that comes in with the base pair. So acyclovir is going to be specific because it requires a virus enzyme to activate it. AZT is a cancer drug, and this is going to inhibit reverse transcriptase. This must be phosphorylated, and even though it has some affinity for our polymerases, it has much higher affinity to HIV reverse transcriptase. So as long as you dose the patient properly, it's going to have minimal effect on host DNA polymerases and have a maximal effect on the HIV reverse transcriptase. Now some other important HIV reverse transcriptase inhibitors are listed here. Dioxyinosine, didioxycytidine are going to all lack that 3' OH group. We have lamivudine, and that's going to have a modified ribose, so the sugar looks different. It's not going to be able to incorporate or bind to the other sugars. They're all going to be inhibitors of chain elongation. Now we also have rhabdovirin. This is going to, or ribavarin. This is going to be an analog of guanosine. It's going to inhibit a lot of things. So when you do messenger RNA, you have a 5' cap, and so those are guanosine caps, and so it's going to inhibit messenger RNA capping, which it makes the message more able to be degraded. It's going to also inhibit the formation of nucleosides. So it's going to deplete your intracellular sources of guanine, and that's through a purine recycling. So this has a little bit of more broad reaction. It doesn't require a virus-specific enzyme necessarily to be activated. So it's going to be active against RSV, Lassa fever, flu or measles, possibly even SARS-CoV-2. There's data that suggests it can, data that suggests it doesn't. And it's all because it's going to interfere with the salvage pathway. Purine, forming purines is very energy, takes a lot of energy. And so if you can prevent recycling of those, the salvage pathway, which is what recycles them, then you're going to really put a big damper in cells that are actively replicating, especially viruses. Now, to target protein synthesis, this is going to be a poor target because our cells need it. And even though interferon is made after a virus infection by our cells to protect our cells against the virus making their own proteins, this could eventually kill our cells. So interferon was treatment for hepatitis C patients in the 90s, and it had some very severe side effects. Protease inhibitors have sort of replaced the interferon, so protease inhibitors are replaced. And they've replaced interferon therapy for hepatitis C. So now instead of interferon, you treat patients with protease inhibitors. However, this is very expensive. So in many areas of the world, interferon is still used. Now, there are also drugs that are going to target assembly and release. So you have HIV that has proteases that are essential for assembly. So you have a series of drugs that are going to target that protease. And so what they do is they're going to be competitive inhibitors because they fit into the active site of the actual protease. So this is a competitive. You also have hepatitis C virus protease inhibitors listed here. Influenza virus neuraminidase inhibitor. This is essential for assembly. And so if you can wipe out neuraminidase, then your virus particles for flu cannot form. And so Tamiflu is an example of that. There's a few others that are going to be inhibitors. And these inhibitors are good against both influenza A and influenza B. Influenza A is the most severe pathogen. And type B has less severe typically, but it's not 100%. So you can have outbreaks caused by B as well. And then to activate the immune response, so stimulation of the immune system is immunotherapies. And it's thought that if you can stimulate the immune system through these immunotherapies, you can sort of get the adaptive response, and especially your T cells, going again. So there's CpG oligodeoxynucleotides that are going to target toll-like receptors. Imiquimod is also going to target toll-like receptors. And this will induce inflammation. Interferon is also going to have an effect on your your protein synthesis. Double-stranded RNA is going to trigger toll-like receptors. And there are several different types of double-stranded RNAs, something called poly-RC, poly-IC. Though all can be added and activate your innate immune response, which in turn can induce inflammation and activate your acquired response. There's also antibodies for passive immunization, and we can add in here SARS-CoV-2. And the antibodies are, of course, are going to act as a neutralizing to neutralize viruses before they can further infect the person.